The human body is a complex machine, which may respond differently to diverse situations, depending on its genetic background, environment, or time. Innovative techniques allow scientists to measure many parameters in human tissues. Thanks to new computational methods that can integrate existing knowledge about how the human body functions, as well as data that comes from these innovative techniques, we can now develop a much deeper understanding of human biology. This approach is called systems biology. Systems biology can be applied to understand disease mechanisms or the mode of action of toxic substances. For instance, if we want to differentiate healthy from sick individuals, we can measure the RNA content of their blood sample and extract specific gene signatures. For example, we can identify a signature set of genes that allow us to distinguish between individuals who have not been exposed to a substance versus those who have been exposed. The ability to find such gene signatures depends on many factors, such as how homogeneous the individuals are within a class, how clear the separation between classes is, and how frequently the classes appear in the population. The population studied is often very small and potentially biased, so it is important to keep in mind that this may prevent the signature being applicable in the general population. So how is a gene signature usually identified? First, we have to define the classes we want to distinguish, such as exposed versus non-exposed. Then, we select subjects from the population who belong to these classes and collect samples of their blood and profile them. As we want to minimize problems occurring when generalizing the signature, we split samples into two data sets. The first one is the training data set and is used to extract the signature. The second one, the test set, is used to estimate the performance of the signature. Following data processing, computational approaches such as machine learning methods are applied on the training set to extract a subset of genes which combined constitute a gene signature, allowing us to distinguish the classes. This signature can then be applied to the test set to estimate the performance of the classification. Extracting good and generalizable signatures isn't easy. Many claim to have found good signatures, but few are approved and used in practice. In fact, scientists assessing their own method often conclude it's the best one. That's why the scientific community needs ways to independently verify methods and results. The SBV Improver project allows this independent assessment by building scientific challenges for methods benchmarking. The first SBV challenge, called Diagnostic Signature, aims to identify specific gene signatures predictive of different diseases. You can still see how well you perform on this task by using the benchmarking tool at www.sbvimprover.com/challenge-1. We've now launched a new computational challenge in the area of systems toxicology to identify markers of the body's response to specific exposure. Every day, humans are exposed to chemicals such as cigarette smoke or pollutants that may trigger molecular changes in their cells. The identification of specific markers of response to this exposure is important to help assess whether a subject has indeed been exposed to particular chemicals or toxicants. After collecting blood samples from exposed and non-exposed subjects, gene expression levels are measured. In this challenge, data related to smoking and cessation status will be used by the participants to answer the question, are gene expression changes measured in blood cells of the subjects sufficiently informative to identify gene signatures capable of classifying them in the correct exposure groups? Rodent data sets will also be provided to evaluate the translatability of the signature across species. You can find a more detailed explanation of the challenge at www.sbvimprover.com. sbvimprover.com/comp-start. So, why should you be part of the SBV Improver Challenge? Become a best performer and present your approach at an international conference. Co-author a scientific article describing the outcome of the challenge. Access new, high-quality data. Receive an independent assessment of your methods. Develop the 21st century predictive systems toxicology. Leverage the wisdom of the crowd. Grow your professional network and enhance your visibility. Be part of 21st century toxicology by joining this challenge. Compete and experience the power of the crowd. To find out more and to take part, visit www.sbvimprover.com/comp-start.